Thank you. So it is my honor to present the lightning talks, and I have to tell you that as our speakers acknowledged very rightly, the work is done by a very large number of many talented graduate students that we have in the department. We have more than 500 of them. So in order to give you a bit more inspiration, we really did save the best for last. And we selected 5 of those students, less than 1%, to give us very brief lightning talks about what they are working on and what they are so passionate about. So I would like to call the first one of them. It is Sangmin Wang with Helen Wills, Neuroscience Institute, and of course the ECS department, to talk about neural network connectivity estimation using optogenetics.
Thank you very much for the introduction. My name is Sangmin, and I'm a fourth-year graduate student in the EECS department here, working with Professor Chunlei Liu. So I'm going to be talking to you about the project titled Neural Network Estimation. I'm talking about real in vivo neural networks, not the artificial ones. And we're going to be using a new technique called optogenetics. And this is a joint work with Ian Oldenburg, who is a postdoc, and Professor Hillel Lesnick, as well as Professor Chunlei Liu. So the main overarching objective of our lab is to develop the tools to study the brain. We still don't have a good understanding of how the brain works. So I'm just going to give you a little bit of overview on what we understand about the brain so far. We have a lot of good understanding about how the brain works at a very micro level. We know the differential equations that describe the action potential dynamics, how the membrane potential of an individual neuron changes and evolves over time. So we have many models to describe this process. And we also have very advanced imaging tools that allows us to study the macro level of the brain. So we have tools like MRI, fMRI that gives you a dynamic contrast images of the brain. We have other contrast mechanisms ranging from diffusion tensor to T1 and T2, all the way to susceptibility tensor imaging to give us an idea of which region of the brain gets activated and it gives us tools to see the brain at different levels. But it is very difficult to image the brain and decode the brain at the same time. So we're relying on a very new technique, this very exciting tool called optogenetics. And in the video you see here, we're going to be targeting a neuron in the middle. You'll see it flicker on and off in green light. We're using a GCAM signature, and that is just a calcium trace indicator, and it kind of tracks the actual membrane potential of a neuron. And when we now have the technology to stimulate a custom ensemble of neurons with cellular resolution and a temporal resolution that's good enough to be able to see the action potentials of an individual neuron, and we can perturb and stimulate an individual neuron in a field of view. And a normal field of view consists of about a voxel size that ranges from around 800 micron to 800 micron with the depths of 100 micron. And we'll be studying the cortical region of the brain where most of the brain computation is carried out. So this is the algorithm that we developed to kind of study the brain and be able to estimate the connectivity among the neurons that we see in this field of view. And this is a typical example of an experiment we do. This might be the resting state of the neurons, where the greener the neuron is, the higher its membrane potential is. So obviously if it's very green, then we can determine for sure that an action potential has fired. So we select a random ensemble of the neurons in the field of view, and we decide to stimulate those and see how the network changes. So overall, again, we have this multiple stimulation profiles that are randomly selected, and we carry on and get this input-output observations. So we developed a deterministic model to describe this population level dynamics that we see at this mesonym level of brain. And we determined a deterministic autoregressive model that's based on the slow decay time of the calcium trace indicator. And we posed it as a system identification problem. So we posed connectivity that is unknown as a system, like a black box system that we're trying to estimate. And we're going to be putting in this random simulation profiles as the input to the system, and the observations, the membrane potentials of all the neurons in the field of view will be our output. And we also know from neurobiology that the connections in the neurons are quite sparse. We expect about 10% sparsity level, and taking advantage of this fact, we apply a modern compressed sensing technique to be able to solve the system identification problem. And we performed some in silico experiments to kind of demonstrate that our algorithm works. And within this center red box field of view, we can see that the estimated network is very similar and it's very close to the theoretically ground truth model that we generate. So the right one is the theoretical one, and the left figure matrix that you see is the estimated one. And this is the example of an in vivo experiment that we did. So you can see the same voxel, and we're simulating random subsets of the neurons, analyzing this data. So the left-hand figure shows the data where x-axis is the time and the y-axis is the neurons, and the red dots indicate where we're stimulating. And the matrix on the right is the recovered matrix. And we'll be basically validating this model very soon. This is a work in progress. And this work has a lot of applications, one of which is that we can immediately start providing some supporting evidences for theories in neuroscience. And obviously, in the near future, this will have very good applications in brain-machine interfaces and medicine as well. Yeah, that's it. Thank you.
Thank you. We will take all the questions for the 5 students at the end. So I would like to move on now to another topic presented by Ali Moin with BWRC and the SORM Lab. The title is Flexible EMG-Based Hand Gesture Recognition, Wearable Device for Controlling Prosthetics.
Thank you. Hi, everyone. I'm Ali Moin. I'm co-advised by Professor Jan Rabai and Elad Alon. And today I'm presenting our flexible EMG-based hand gesture recognition wearable device for controlling prosthetics. So gesture recognition for human-machine interfaces is basically trying to improve the way that humans interact with machines. For instance, it provides more natural inputs to games, artificial augmented reality, virtual reality systems, or as another example, it provides more natural control for prosthetics and rehabilitation devices. In order to do this gesture recognition, we need to look at the signals, biosignals in the body that reflect the muscle movements and contractions. One of these signals is called electromyography, or EMG, which can be recorded non-invasively from the surface of the skin. So if you look at this animation, you can see the action potential coming through the nerve, and then when it reaches to the muscle fiber, it causes the muscle to release neurotransmitters and cause the muscle to contract. So if we put an electrode on the surface of the skin, we can basically record this electrical activity and feed it to a classifier to recognize the gestures. Well, if you look at the state-of-the-art EMG systems, most of them are either commercial high-end systems that are very bulky and expensive, or the ones that are in wearable form factor have very low number of channels and few gestures to be recognized. If you look at the research community work, they're still very bulky, they are not wireless and not comfortable to use as an everyday wearable device. So what we try to do is to have a miniaturized device that has a high number of electrode channels, be wireless, and very comfortable to wear as an everyday wearable device. And on top of all these systems only record the signal and then do the processing, learning, and classification either offline or on a PC. What we are targeting here is to do the whole learning and classification on board. So these are the different blocks of our system. We start with printing a flexible sensory array. It basically screen printed on a flexible substrate in Professor Anna Arias' group in our department. Then we designed a very small PCB that has custom chips for recording and digitizing the EMG signal. And also it has an onboard FPGA, which we have implemented our learning and classification algorithm called hyperdimensional computing, all on this FPGA. And we use the PC only to show the user to do the sequence of gestures for training, and then we can do classification and everything on board, and then transmit the gesture to the actuator, either being a prosthetic arm or a robotic arm and so on. So far, we have shown classification of 21 gestures, including all individual fingers, flexions and extensions, counting numbers, and so on. So I have a short video here. So we start by the printing process. This is silver ink. We put it on a flexible PN substrate, and it goes through the screen printer. And what you get out is basically an array of 4 by 16, 64 electrode arrays, and the traces. Then we attach our PCB, the neural acquisition front end, to this electrode array. And we add a drop of hydrogel on top of each electrode just to maintain a better contact with the skin. Then we wrap the electrode array around the arm. And then we ask the user to do a sequence of gestures. We only require to do each gesture once for training, which is one of the advantages of our learning algorithm, with very minimum amount of data. And then we can do online and onboard classification of the gestures with very low power and low latency. We have shown multiple demos using this device. For instance, we can control a robot or a robotic arm. And we even went further and 3D printed a prosthetic arm, so we were able to control a prosthetic arm mimicking our gestures. So if you are interested in this research or other projects in our lab, please come visit us at our open house, 1 to 3 p.m. at Swarm Lab, 490 Corey Hall.
Thank you. The next talk is going to be given by Melinda Perkins. The talk is about biological networking and how electrical engineering meets regenerative medicine.
Thank you for the introduction. Hello, my name is Mindy Perkins, and I am a fourth-year PhD student working with Murat Arcak. When I tell people that I research biological patterning, a question I often receive is, what does that have to do with electrical engineering? Well, from one perspective, it's all about networking, how individual units coordinate their activities. Electrical engineers have developed a number of useful tools and techniques for analyzing networked systems. You could think of computers on the internet, or cars on the highway, or biological cells in the body. Network-level group activities are especially important for organisms because they may possess millions of cells that need to coordinate what they're doing in order for the organism to function properly. And one especially important time in which activity must be spatially located in the appropriate place occurs during embryonic development. Understanding the principles behind embryonic development and spatial patterning at that time are important for scientists working in regenerative medicine because they aim to grow tissues and organs to replace or repair damaged ones in patients. In natural systems, tissues develop through a complicated, elaborate series of processes in which individual cells must determine which genes they are expressing. Depending on the genes that a cell expresses, it will adopt a different fate in the adult organism, becoming, for example, a skin cell or a neuron. Because the spatial location of different cell types is very important for the proper functioning of a tissue or an organism, early spatial patterns of gene expression are essential for appropriate development. But how do initially identical populations of cells spontaneously produce gene expression patterns that vary in space by cell? Actually, the early computer scientist Alan Turing was one of the first people to investigate this problem. His reaction diffusion theory of patterning has been highly influential in biological research in the past 70 years. It has been evoked to explain the development of spots and stripes on animal coats, as well as the early stages of the formation of fingers and toes. While Turing's theory is very powerful and insightful, it also has a number of constraints that are difficult to satisfy in biological systems. In other words, many patterns may look like Turing patterns without following a Turing mechanism. In my research, I aim to look at how spontaneous patterns may arise in systems that do not necessarily satisfy Turing's criteria. To do so, I model individual cells as input-output systems. They receive inputs from the environment and from other cells. These signals may be electrical or chemical, mechanical, or some sort of sensor like temperature. The cells then produce outputs. They produce signals that go to their neighboring cells, and they also have some level of gene expression that is influenced by the inputs that they receive. Individual input-output systems of cells can then be hooked together into a network, and that network structure can be analyzed using tools from control systems and signal processing. In my research, I aim not just to develop theory, but also to apply my theory in practice. To that end, I have been collaborating with labs here at Berkeley as well as abroad to generate gene expression patterning systems in bacteria and in yeast. Studying these systems in microorganisms is the first step towards being able to implement them in human systems, so that one day, patients who have damaged organs and tissues will not have to wait for a donor to receive a replacement. Thank you.
Thank you, Midi. I would now like to ask Roshan to come to tell us about learning to fold, machine learning for protein structure prediction and biochemistry.
Hello. I'm Roshan Rao. I'm a second-year PhD student in CS. So today I'm going to talk about some work that I've been doing in collaboration with Peter Chen, Rocky Dwan, and Peter Abil on how to use machine learning for protein structure prediction and protein folding. So I'm going to talk about a little bit at a high level about what protein folding is and how we use machine learning to do this. And I'm also going to touch on something that some of you might have heard of, which is DeepMind's AlphaFold, which recently beat state-of-the-art on this task, and what approach they use to solve this problem. So the first thing is a bit of an overview on what a protein is. So a protein is a sequence of amino acids, and this is an example of a simplified amino acid, where each amino acid is bonded to the next and previous amino acid in the sequence. So for those of you who are somewhat familiar with machine learning techniques, you might kind of automatically recognize this as something that we might want to apply sequence learning to. So techniques from natural language processing or from audio processing are kind of the things that we're going to look to try and solve this problem. So what's the protein folding problem? What are we trying to predict? So, you know, we can go out into the lab and determine a lot of information about proteins. So one thing we might want to do is go out and determine the amino acid sequence of a protein, the coevolutionary data, the sequence alignment data of a protein. And this is all kind of stuff that or an experimenter can go out and determine pretty easily. All right, so that's all good. But the stuff that you actually want to know in order to make predictions, in order to design drugs, and in order to actually have this be useful are protein structure, protein function, protein interaction. So, you know, what are these proteins doing? And this stuff, it turns out, is really, really hard to determine in the lab. So on the order of months or maybe even years of effort by a biochemist and in the lab. So the protein folding problem says, okay, let's say I go out and find the stuff that's really easy to find. So I'll experimentally determine the sequence, the coevolutionary data, and the sequence alignment data. Can I then take all of that stuff that I learned, feed it to my computer, and have it predict the structure, the function, and the interaction, and all of the stuff that's hard, but that I actually really care about? And if we do that, then everyone's happy. All right? So where does ML come in, and why have we been seeing kind of increased success in protein folding through machine learning? Well, one big thing is that, you know, data sets are becoming bigger. So this is a graph of the CASP competition, which each year the data set of each CASP is a superset of the CASP of the previous year. So they experimentally determine more proteins, and they add that to the current data set. And each year, both the data set is getting bigger and the techniques are getting better, and we're just seeing better and better improvement in performance. And you can kind of see at the very end, there's a spike between first and second place, which is AlphaFold and DeepMind's result. And a lot of these results are coming because not only through bigger data sets, but also through better techniques. So we have things like residual networks and convolutional networks, which have been introduced through vision and language, and we're able to leverage those to determine, have better and better techniques for predicting these things. So in our approach, we're going to use self-attention networks, which have seen a lot of success in sequence-to-sequence natural language processing tasks. So this is an example of self-attention applied to language, where each word can see every word in the input. And our idea is that if each amino acid can kind of look and pay attention to every other amino acid, then you can learn better and better global relationships between different amino acids. And so their DeepMind's recent approach also follows a pretty similar general structure. So they're going to use convolutional networks and residual networks, and they have slightly better data. They have a better objective function that they optimize than we currently do. But a lot of these things combined really formed really excellent progress in the field. And so lastly, I want to end on where I see this field going in the future and what some next progress is. So I think there's still plenty of room for improvement. I don't think this is a solved task by any means. And here are just a handful of things that we can still borrow from other fields. So natural language processing has seen character and word level embeddings help a lot. Leveraging unsupervised learning in natural language processing has also helped a lot. And a lot of these things can then also be applied to other problems like protein docking, protein function prediction, and actually designing proteins from scratch. And yeah, that's all I have. Thank you.
Thank you, Roshan. I would like now to welcome Margaret Chapman, who's going to tell us about breast cancer treatment as a dynamic control problem.
Thank you. My name is Margaret Chapman, and I'm a PhD candidate in electrical engineering advised by Claire Tomlin, and I will present our work on breast cancer treatment as a dynamic control system. These are images of triple-negative breast cancer, or TNBC, cells following 72 hours of treatment with increasing doses of different drugs. TNBC is a particularly aggressive and deadly form of breast cancer that is more common in Black women and in younger women. It tests negative for the 3 most common proteins in breast cancer. So therapies that target these proteins are not effective. Also, triple-negative breast cancer cell populations are composed of a diverse mixture of cells. So it's very difficult to treat all of these cells at the same time with a single drug. In this picture, you see green cells, red cells, and blue cells. These cells glow different colors because they have a marker that's attached to their nucleus that reflects the set of phenotypic traits that the cells exhibit. And in particular, we find that these cells, these different cell types, respond distinctly to different drugs. I'm going to explain how we use models to better understand this dynamic system and work towards controlling this dynamic system. Using a modest amount of time series data, we identified models of the evolution of triple-negative breast cancer cells following initial treatment to a particular drug. The models assume that the live cells occupy a particular phenotypic state and can transition, divide, or die. I would like to provide a snapshot of one biological insight from our models. An important scientific question is to understand why the green cells are able to flourish under the drug trametinib. This is an important question because the survival of the green cells is able to bring the cancer back. Parameter values, uncertainty analysis of parameter variability, and model validation on separate data indicate that green cells may be able to be enhanced under the drug trametinib due to increased transition from blue to green and reduced transition from green to red. So a natural treatment strategy would be to steer the population to the green state using trametinib and then kill the more homogeneous population with an effective drug. The problem is we don't have a drug that kills the green phenotypic state. More generally, it is very difficult to detect the sensitivities of particular phenotypic states to particular drugs, and it's very difficult to identify effective drug schedules by empirical means alone because there's so many combinations to test. Consequently, we studied the drug scheduling problem from a more abstract mathematical perspective. We could start with a collection of drugs at a single dose. Some of these drugs are more harmful to healthy and cancer cells, and other drugs are less harmful. We wanted to see if it was possible to identify a sequence of drugs that was tolerable but still managed to cause the cancer to die over time. So we quantified tolerability of the drug schedule in terms of longer waiting times between treatments because this corresponds to reduced total drug concentrations to be applied over a given time interval. In the ideal scenario where we assumed perfect models and in each cycle, every drug would be applied once, we designed an algorithm that optimized for longer waiting times between treatments. And using the output from this algorithm and the knowledge that our models are actually imperfect, we developed mathematical conditions to guarantee cell death over time. And here's an example simulation of these cancer cells dying over time under our imperfect models and our mathematical conditions. In the future, we look forward to augmenting our mathematical rules with additional data that is being collected by our collaborators at the Oregon Health and Science University. Thank you so much, and I appreciate any questions.
I would like to thank all our student speakers and thank you all for your attention. Given that we are running a little bit late, I would like to recommend that we interact with the students with questions as we mingled over lunch.